So when we think about research in the area of eosinophilic GI diseases, I think there's a couple important points to consider. The first is that research is actually repeating or re-examining search. And we are, in many ways, searching for many of the answers to the questions that have been posed. They haven't been addressed to date. For instance, what are abnormal numbers of eosinophils in the stomach, small intestine, and colon? We haven't truly defined that yet. So there are a number of questions that we're trying to examine for the first time, and it is search as much as it is research. When we think about research, there are a couple different ways to think about this. And the first is, I'd say that we are undergoing a rapid period of increasing awareness of this disease. So much of the research that's been done has been to say, what are the symptoms associated with this disease? Many of the clinical studies have identified the fact that younger children can have very common symptoms associated with reflux disease, abdominal pain, vomiting, things that are relatively commonplace that have been thought to be due to other problems that in fact are due to eosinophilic esophagitis. One very new and rapidly increasing area are feeding dysfunction. So young children can have problems eating. They may not eat as much as we think they should. They may avoid certain foods. And in fact, that can be one of the presentations for young children who have eosinophilic esophagitis. For older patients or adolescents and adults, some of the key features are food impactions or food getting stuck in the esophagus, not going down as well as it should, chest pain, or problems with swallowing. Those three features really help to characterize the adult patterns of this disease, but they have not been quantified to date and now are undergoing definition. So awareness of this is quite important. The second feature of detection or recognition of symptoms is the coping features of this disease, so that many patients may have already developed ways to avoid the symptoms. And what do I mean by that? If I were to ask you, do you have problems swallowing? You might say no. But if I asked you, do you drink water with your food? Do you take excessively long time or longer than your family or friends to eat food? Do you chew your food a lot? Do you pocket food in your mouth? Use liquids or gravies or butters to lubricate the food to go down, or avoid certain foods that are highly textured, such as meats or breads? The answer may be yes to those, and those may not become manifest unless you ask more than one question. That's one area of research that I think is very important, just to say, who are the patients and what kinds of symptoms do they have? The second area that we think about are kind of do we know about the genes and what may predispose someone to having this disease? How do we measure the disease itself from a functional standpoint? And the third is, how are we going to take care of patients who have this problem? We think about the things that may predispose someone to this disease or lead to the actual problems that we incur. This is a very active area of investigation, looking at genes and proteins that the body may produce that cause the symptoms to happen. In that regard, there have been a number of genes that have been associated with this disease that include genes that may cause more eosinophils to go to the tissue, that may cause the barrier itself within the esophagus not to function as well as it should, and abnormalities of the immune system that may predispose other cells to come that perpetuate this inflammation. When we think about ways to understand this disease and what it's causing, there are a number of new techniques and devices that may help us understand the function and ultimately lead to the diagnosis. An endoflip is a new type of technique that helps us measure the stretchability of the esophagus, or how fibrotic or scarred it might be. There are motility monitors that can do the same kind of thing, looking at motility disturbances. There are pH impedance tests that help us understand how much reflux may or may not be a part of this disease. We're working on some novel technology to understand some minimally invasive ways to measure inflammation, something called the esophageal string test or esophageal inflammation monitor, in which a capsule that's filled with a string can be swallowed. That string leaves a trailing edge within the esophagus that can capture inflammation molecules that may help us avoid endoscopy and biopsy in the future. And then when we think about forward parts of this, how can we take care of our patients? And I think all of the things that we do within research want to have some kind of clinical applicability, there are a number of new molecules now that are being developed, specifically relating back to some of the genes and proteins that have been identified now. So there are antibodies that may target some of those molecules, including anti-IL-5, anti-IL-13, anti-eotaxin 3 molecules that may help us understand that better. And there are other thoughts now of ways to prevent these reactions from happening. So I think in the sense of trying to target new therapeutic molecules is the way of the future.